Hello everyone, at this point in our discussion, we will be talking about the different characteristics of stem cells. So what makes stem cells unique and powerful? The very first reason is that stem cells are capable of symmetric and asymmetric cell division. So symmetric cell division, this refers to the usual cell division nga itong nahibalaan. So example no kay, kanang mahitabo sa mga epithelial cells. So from a mother epithelial cells, if it will divide, maka-form siya og two epithelial cells. So meaning that the mother cell and the daughter cell both have the same fate. Meanwhile, asymmetric cell division, ganito ang nakalahi gyud sa ato ang stem cells because stem cells are the only cells that are capable of asymmetric cell division. Asymmetric cell division refers to the cell division wherein the two daughter cells have different fates or lahi sila og future. So for example, if this is our stem cell, the mother stem cell, and habang nagamitosis siya, mo niya mga daughter cells. So the first daughter cell can become committed to a certain cell line. So for example, this daughter cell naka-decide siya nga mahimo siyang common lymphoid progenitor. Meaning, kani siya nga from this cell, pwede mag-emerge ang mga lymphocytes. At the same time, the other daughter cell, pwede siya dili mahimong common lymphoid progenitor. Pwede siya mahimong stem cell ug balik. Pwede siya mutundog sa iyahang mama. So that is asymmetric cell division. Lahi og fates or lahi og future ang two daughter cells produced. Next is, stem cells are capable of cell renewal. Stem cells are very tough cells, no? Once damaged, they can easily heal themselves, and at the same time, they are capable of self-renewal and self-repopulation. You know, the rest nasa spelling. So, stem cells are capable of self-repopulation because they are capable of asymmetric cell division. So, maskin habang naga-divide sila to produce some daughter cells that will become blood cells, other daughter cells can become stem cells. Meaning, kaya sa stem cells na i-repopulate iyahang self as stem cells. Stem cells can give rise to differentiated progeny, meaning that if a single stem cell will undergo series of cell maturation, cell differentiation, or cell processes, it will produce cells that are of different types. So for example, this stem cell, once mag-undergo na siya og many processes, many changes, it will produce an eosinophil, basophil, or neutrophil. So these three cells are different. However, they came from a single stem cell. So, mo to siya ang meaning sa stem cells can give rise to differentiated progeny. Next, stem cells are able to reconstitute the hematopoietic system of an irradiated host. So, this characteristic of stem cells is very evident in the experiment done by Till and Makalok. So, in that experiment, they irradiated the spleen and the bone marrow of a mouse. So, diba, we all know pag mag-irradiate or pag i-expose natin ang isa ka organ sa high levels of radiation, the cells in that organ will die. So, katong mga hematopoietic stem cells aning bone marrow ug sa spleen, nangamatay na to sila. So, devoid of hematopoietic cells or devoid of cells na ni sila na mga organ. So, in that irradiated mice, they injected marrow cells from a healthy mouse. So, nag-inject sila og mga marrow cells from a healthy mouse. They found out 7 to 8 days later na ang katong irradiated mice was able to form or develop a hematopoietic system from that marrow cells alone. Tapos katong mga marrow cells are composed mainly of hematopoietic stem cells. So, from that experiment, they concluded that hematopoietic stem cells can produce or form or build the entire hematopoietic system of an irradiated host. So, in ana sila ka-powerful, no? Because despite of the fact na wala na gyud cell ato nga mga organs, they were able to build or form or produce the different lineages of blood cells, or they can produce the ancestors of the different blood cells present in the circulation. Another characteristic of stem cells that is also common to most cells in the body is that it is capable of doing apoptosis or the programmed cell death. So once the stem cells sense that the damages in it is not repairable anymore, it will do apoptosis in order to prevent the damages, especially damages sa DNA, to be passed down to its daughter cells. If ma-pass down man gud ang damaged DNA sa mga daughter cells, of course, meaning damaged po ang DNA sa daughter cells, and it is not good for the body. Another characteristic of stem cells is that it is stuck on the GO or the G0 phase of the cell cycle, also termed as the quiescent stage. So, di ba remember, kapag naa sa quiescent stage or quiescent phase ang inyong cells, meaning metabolically inactive na sila, meaning wala kayo cellular metabolism or cellular activity nga mahitabo sa inyong cell ana. So, that is done in order for the stem cell to defend itself, to defend its DNA from possible damages. Because when the cell is metabolically active, active po ang iyahang DNA. And once active ang iyahang DNA, mas prone ang DNA to damages. However, if the stem cells decide na gusto niya mag-undergo og mitosis, muhawa siya sa metabolically inactive na phase. Mag-adto siya og balik sa G1 nga phase. Remember these phases sa inyong cytogen? So, dili po na dali-dali no nga pag after niya og G0 na phase or after niya quiescent na stage kay decide niya na gusto niya mag-proceed to the G1 phase kay gora na dayon. So, it's not like that. Napay transitory phase. The transitory phase or the phase wherein the cell will prepare itself to go back to its cellular metabolism or to its metabolically active state. So based from these characteristics of stem cells, we can conclude that stem cells have three fates or three possible futures. First is that stem cells can opt for self-renewal or self-repopulation, wherein si stem cells, pwede niya i-decide nga, dili lang siya mag-cell differentiation, dili lang niya gusto mahimong blood cell, gusto lang niya mag-stay as stem cells. Second fate or second possible future is that mag-undergo siya og cell differentiation. Pilion ni stem cells na mahimo siyang blood cell. And the third possible fate and the most sad fate of all is pilion ni stem cell na mag-undergo og apoptosis. So this is the possible fate or the possible future of stem cells if ever daghan kaayo og mga damages na na-acquire ang iyahang DNA. In order to prevent the damages to be passed on to the next generation of cells, the stem cells will decide to kill itself. Joke lang. To do programmed cell death. So what are the factors that influence or unsa kaya ang mag-ingon sa imong stem cells na dapat magstay lang kang as stem cells or dapat mahimo kang lymphocyte, dapat mahimo kang RBC? So our scientists have developed models, the stochastic model and the instructive model, to serve as explanations about the mechanisms undergone by the stem cells in order for it to decide its future. First is the stochastic model. So the stochastic model tells us that the stem cells decide its future randomly. So depende na lang random lang yun, no? Depende na lang kay stem cells kung unsa ang iyahang pilion, self-renewal or cell differentiation. Now, if pilion niya ang cell differentiation, depende na lang pod sa iyahang innate characteristics, depende na lang pod sa transcription factors na abundant sa iyaha, kung unsa nga cell line ang iyahang ma-decide. So, the stochastic model is also termed as the intrinsic regulation because parehas atong na-mention ganina, it is up to the cell itself. So, innate na gyud or natural na lang gyud sa stem cell daw kung unsa nga bloodline ang iyahang pilion depending on the transcription factors na present sa iyaha. So remember what are transcription factors? Transcription factors are the gene regulators. So sila to ang mga naga-decide kung unsa nga gene ang ilang i-express or unsa nga gene ang i-suppress, therefore telling the cell what to become. Now the instructive model tells us that the future of the stem cells is determined by the bone marrow microenvironment. So the instructive model is also termed as the extrinsic regulation because dili daw na up to the stem cell itself or innate, dili daw na innate sa stem cell kung unsa ang iyang future na magustuhan. Depende daw na siya sa iyahang outside environment, which is the bone marrow. So depende daw na siya sa mga cytokines, sa mga proteins released by the different cells in the bone marrow to influence the stem cell to become what it is. Kung mag-differentiate ba siya or mag-self-renewal, or if ever mag-differentiate siya, unsa bang iyahang gusto maging? Gusto ba niya maging RBC, maging lymphocyte, or maging neutrophil? So, modern approaches tell us that dili daw na solely stochastic or dili daw na solely dependent lang sa stem cell itself. Dili po daw na solely instructive or dili daw na solely dependent lang sa mga external environment. It is dependent on both because the external factors will not have an effect to the cells if the cells do not have the transcription factors needed for it to respond to the external stimuli na gina-direct or gina-hatag or gina-influence sa external environment sa iyaha. So examples no kay, let's just have a very summarized example. The bone morphogenetic protein 4. So bone morphogenetic protein 4 together with other hemocytopoietic or hemopoietic cytokines will influence the stem cells in the bone marrow to produce or to undergo cell differentiation and not cell renewal. So si BMP4 and other hemopoietic cytokines will tell the hematopoietic stem cells to undergo cell differentiation. But dili maka-respond si HSC or si hematopoietic stem cell sa kani nga mga proteins if wala siya atong transcription factor na SCL. SCL stands for stem cell leukemia. So this is a transcription factor, no? Without this, dili siya maka-respond kay BMP4 and other cytokines to go or to proceed to cell differentiation. So this is the chart summarizing the entire hematopoiesis. So don't worry, natay separate discussions for erythropoiesis, for leukopoiesis, and for megakaryopoiesis. As for now, summary na lang sa gyud ta. So from a multipotent progenitor hematopoietic stem cell, there arises common myeloid progenitor and common lymphoid progenitor. So sa common myeloid progenitor, dinhi gikan ang mga neutrophil, monocytes, eosinophil, basophil, erythrocytes, and megakaryocyte. So, as you can observe, special good ang mga lymphocytes because they have a different progenitor. Unlike sa RBC, platelets, and granulocytes na naay common myeloid progenitor. So, without further ado, let us begin. Ang sinugdanan gud sa ilahang tanan kay si CFUS or colony forming unit stem cells. So, from the stem cells, from the multipotential stem cells, there arises the common lymphoid progenitor and the common myeloid progenitor. The common lymphoid progenitor is specifically known as the colony forming unit lymphoid or the lymphoid colony forming unit. So, ani gid na ang iyahang scientific notation, ang iyahang pagkasulat scientifically, CFUL. And the common myeloid progenitor is the CFU colony forming unit, granulocyte, erythrocyte, monocyte, and megakaryocyte. Now, let's proceed to the erythropoietic lineage. So, ang madiscuss karon is kutob lang sa mga precursors. Ay, progenitor, sorry. Atong madiscuss karon kay kutob lang sa mga progenitors. So, mga progenitors, mauni silang katong mga na-IU sa last. Like, CFUE, BFUE, ganun. Naapod tayo mga precursors. After sa progenitors are the precursors. So, precursors, katuning madunggan ninyo nga mga blast, no? Erythroblast, megakaryoblast. So, kato sila. For this discussion, kutob lang ta sa progenitors. Kaya naman po jud tayo separate discussion for those precursors. So, let's begin with the erythropoietic lineage. So, from the common myeloid progenitor, si CFU-GEMM, there arises the BFUE or the burst-forming unit erythroid. So, si CFU-GEMM will become BFUE in the increased presence of erythropoietin, interleukin 3, and GM-CSF. So, unsa ni sila si interleukin 3, erythropoietin, and GM-CSF or granulocyte, monocyte, colony stimulating factor. So, these three cytokines will influence the CFU-GEMM to become BFUE or the burst-forming erythroid. So, kani si burst-forming erythroid, committed na ni siya no, to become erythrocytes. May pa siya committed. So, what's different? Si BFUE, since committed naman siya to become an erythrocyte, immature pa ni siya kaayo no? Immature kayo siya in a way nga baba pa ang iyahang erythropoietin receptors. Since baba pa ang iyahang erythropoietin receptors, dili pa kaayo siya maka further undergo og cell differentiation para ma-classify gyud siya as erythrocyte. So, moto, with the help of these three cytokines again, si BFUE will mature to become CFUE, colony-forming unit erythroid. So, si colony-forming unit erythroid, mo ni siya ang pinaka-mature na erythrocyte progenitor. So, CFUE, daghan na siya, increase na ang iyahang erythropoietin receptors. Mas responsive na siya sa erythropoietin. Now, let's proceed to the megakaryopoietic lineage. So, again, from the multipotential stem cell, arises the common myeloid progenitor. Next is the burst-forming unit megakaryoblast. So, si burst-forming unit megakaryoblast, kay mao na ni siya ang progenitor na committed. Mao na ni siya ang progenitor na committed towards becoming a megakaryocyte. So, after the burst-forming unit megakaryoblast, arises the colony-forming unit megakaryoblast. So, naapanay last progenitor cells, no, ang atong megakaryocytes, that is LDCFU or the light density colony-forming the light density colony-forming unit megakaryoblast. So, what is the difference of this LDCFU meg from BFU meg and CFU meg? So, si LDCFU meg is less mitotically active. So, meaning ang kaning first two precursors ni megakaryocyte, si BFU meg ug si CFU meg, kay very mitotically active. Tapos si LDCFU meg kay less mitotically active, meaning dili kayo siya naga-undergo og cell division. At the same time, sa LDCFUE mag-start ang ginatawag nato na endomitosis. So, sa mga na-under sa ko sa cytogen sa una, na-mention nato ni si endocytosis once sa isa ka quiz, no? So, endomitosis refers to the chromosomal division without nuclear and cytoplasmic division. So, basically, sa endomitosis, nagadivide, nagadaghan ang imuhang chromosomes, pero ang cell kay wala nag-divide. Ang nucleus wala nag-divide. Yes, therefore, it is safe to say na ang megakaryocyte, katong mature form na ani nilang tanan, is polyploidic. Diba, pag mag-ingon ta og polyploidic or polyploidy, Meaning ana kay ang iyahang chromosome number is greater than 46 and multiple of 46. So, for example, ang kaning nga megakaryocyte kay na ay 92 ka chromosomes. That is because, again, ang mga megakaryocytes kay naay capacity nga mag-undergo og endomitosis, and endomitosis begins at light density colony forming unit megakaryoblast. So, ang katong endomitosis refers to the process of chromosomal divisions or managhan ang chromosomes without nuclear division and without cytoplasmic division. So that is it for the megakaryopoetic lineage, the megakaryopoetic progenitors. By the way, wala sad na ako naingon, no? Sorry. Ang difference sa mga progenitors and precursors is that tanang progenitors, let's go back. Kani sila tanan. Tanang mga progenitors, pareha lang na sila naong tanan. They are microscopically indistinguishable. Pareha sila naong tanan sa microscope. They look like large lymphocytes nga dagko kaayo og nucleus and dako og size sa cell and very basophilic. So, meanwhile ang mga precursors katong giingon nako ganina na ay mga blast sa sumpay sa pangalan, mga erythroblast, megakaryoblast kana. Kato sila kay magsugod na og differentiate ang ilahang cells. Magsugod na ang ilahang cells na ma-differentiate na to microscopically like si pronormoblast na siya characteristic sa pagiging erythrocyte. Maklasify na nato siya from other cells. Si megakaryoblast na pod siya characteristics of being a megakaryocyte and ma-differentiate na nato siya from other cells. So again, progenitors are microscopically indistinguishable and they all look like lymphocytes. So let's proceed to the monopoetic lineage. So the monopoetic lineage, again, gikan gihapon sa common lymphoid progenitor nga si common myeloid progenitor nga si CFU-GEMM. So from that CFU-GEMM, once it is acted upon by many cytokines, specifically si GM, CSF, or katong na-mention na ito ganina, si granulocyte, monocyte, colony stimulation factor. Once CFU-GEMM is acted upon by this, it will produce CFU or it will become CFU-GM, colony forming unit, granulocyte, monocyte. So if CFU-GM is acted upon by monocyte, colony stimulation factor, si CFU-GM or colony forming unit, granulocyte, monocyte, once acted upon by monocyte, colony stimulation factor, will become CFUM or colony forming unit, monocyte. So si CFUM, mao ni siya ang last progenitor stage ni monocyte, and at the same time siya pod ang beginning sa monocyte commitment. So kani siya nga progenitor ang committed to become monocyte. Next is the granulopoetic lineage. So same lang kay monocyte, no? After CFU-GEMM, once acted by GM, CSF, and other numerous cytokines na dili na lang jud nato i-mention in this discussion because they are too many, the CFU-GEMM will become CFU-GM. Then, once acted upon by GCSF, the granulocyte colony stimulation factor, CFU-GM will become colony forming unit granulocyte. So, kani siya nga progenitor will be the first progenitor to become committed to become a neutrophil. Yeah, basta kani siya kay mahimong neutrophil. Alam niyo naman siguro spelling ng neutrophil. Next is the eosinophilic lineage. So, eosinophilic lineage kay mas shorter, no, mas gagmay ang iyahang progenitor, duha lang kabuok, iyahang progenitor, CFU-GEMM or the common myeloid progenitor, once acted upon by many cytokines, especially interleukin 3 and interleukin 5, the CFU-GEMM will become CFU-eosinophil or the colony-forming unit eosinophil. Now, this colony-forming unit eosinophil is the first progenitor that will be committed towards becoming an eosinophil. Next is the basophilic lineage. So, just like the eosinophilic lineage, gamay lang na progenitors ang present sa atong basophilic lineage. From the common myeloid progenitor, once acted upon by many cytokines and especially the cytokine interleukin 3, interleukin 4, the CFU-GMM or the common myeloid progenitor will become CFU basophil, the colony-forming unit basophil. So those are the basophilic progenitors. Now let's proceed to the lymphopoietic lineage. So the hematopoietic stem cell, once acted upon by many cytokines, especially interleukin 7 and stem cell factor, will become or will differentiate to become the common lymphoid progenitor or the colony-forming unit lymphoid. The colony-forming unit lymphoid will further differentiate to become either the pre-B cell or the pre-T cell. Then this pre-B cell will become B cell. The pre-T cell will become or will mature to become T cell. So, B cell, once in the lymph nodes or in the secondary lymphoid organs and exposed to different antigens, will become or will further mature to plasma cell. Si plasma cell kay katunan ni siyang cell nga naga-produce og antibodies. Next, T cell, once trained in the thymus, will become either CD4 positive, the T helper cell, or CD8 positive, the T cytotoxic cell. So while we are discussing the maturation of the different progenitors of the different cell lineages, we have discussed or we have mentioned cytokines. So what are cytokines? Cytokines are a wide group of proteins, specifically glycoproteins, that are produced by the immune cells na naay effect sa other cells. So cytokines are produced by immune cells. Then these proteins, the cytokines, will promote, they will influence the cells na ilahang gina-act upon to do something. So cytokines regulate many bodily processes such as hematopoiesis, inflammation, fever. So daghan jud og gamit si cytokines. And in hematopoiesis, they play a role in the proliferation, differentiation, and maturation of hematopoietic precursor cells. Examples of cytokines are interleukins. Interleukins are the type of cytokines produced mainly by leukocyte. So ang leukocyte no nga main producer sa daghan nga mga interleukins is si CD4 positive T cell or si T helper cell. So T helper cell is the major producer of many interleukins. So interleukins, however, are not solely produced by leukocytes, mainly by leukocytes. Interleukins are also produced by they are also produced in small amounts by endothelial cells or fibroblasts. Now, lymphokines, from the word lymphokines are produced by lymphocytes. Monokines, from the word mono, are produced by monocytes and macrophage. So, examples of monokines are interleukin 1 and TNF-alpha, also known as cachexin. I think na-mention man po jud ni sa inyong histopath. So, diba si interleukin 1 is an interleukin and at the same time, it is a monokine. Kaya ginaproduce man siya ni monocyte. So, interleukin 1, kato ni siyang interleukin which plays a role in fever. Because interleukin 1 is a pyrogen, meaning an agent that causes a rise in the temperature. So, those are examples of monokines, interleukin 1 and tumor necrosis factor alpha, also known as cachexin. Chemokines, on the other hand, are cytokines which play a role in attracting WBCs. So, examples na ay example na bacteria din hiya. So, ang mga chemokines will go to this site of infection, and these chemokines will attract the neutrophils para maadto si neutrophil dito sa mga bacteria para ma-engulf na ni neutrophil. Next, interferons. Interferons are also produced solely by, not solely, mostly. Interferons are mostly produced by lymphocytes. So, maklasify nato si interferon as a lymphokine. Interferons play a role in viral infection. So, interferon destroy viruses. Colony-stimulating factors, on the other hand, mao gyud ni sila ang cytokines that play a great role in hematopoiesis. Colony-stimulating factors are classes of cytokines which act mainly on hematopoietic stem cells. So, CSF or colony-stimulating factors, they influence the hematopoietic stem cells to undergo changes such as differentiation, maturation, proliferation in order for the hematopoietic stem cells to produce blood cells. Colony-stimulating factors are produced by many cells in the bone marrow such as the endothelial cells, adipocytes, fibroblasts, and the like. So, these colony-stimulating factors, just like other cytokines, are highly specific. Pero, their name or their target cells is not limited to their name. Meaning, for example, si GCSF, diba, iyahang name stands for granulocyte colony-stimulating factor. Ang katong si GCSF, ang iyang target cells is not limited to granulocytes alone. Iyang target cells are the progenitor cells of many blood cells. However, ang iyahang primary target kay si granulocyte. Colony-stimulating factors are potent, meaning they are active at low concentrations, and CSF also have synergistic effect. So when we say synergistic effect, meaning it can act with other substances in order for it to have an increased effect. So example na kay si interleukin 3, it can act with GCSF for them to further stimulate exponentially the megakaryocyte colony. So that is for colony-stimulating factors. Next, classes of cytokines that are very involved in hematopoiesis are the interleukins. So interleukins, as what I have said earlier, are cytokines produced mainly by leukocytes. However, dili lang siya limited sa leukocytes. Fibroblasts and endothelial cells can also produce interleukins. Interleukins are numbered in order of identification. So dati interleukins are named according to their function. However, they've learned that ang mga interleukins kay dili sila limited to one function only. Ang interleukin nga maka-stimulate of lymphocyte are also learned to have an impact or to have an effect on neutrophil stimulation. So dili lang limited si interleukins, varied pod ang iyahang effect. Therefore, they've realized na i-name na lang nila ang interleukin based in the order of identification. Interleukins are proteins that exhibit they exhibit multiple biologic activities. So, dili lang limited sa isa ka function ng ilahang kaya. Just like CSF or colony-stimulating factors, interleukins also act synergistically with other cytokines. Again, in order to increase the effect exponentially. Interleukins are also potent, meaning they are effective at very low concentrations. We have three types of hematopoiesis. First is the erythropoiesis. So, erythro meaning red, and poiesis meaning formation. So, erythropoiesis refers to the production or the formation of red blood cells. Lipopoiesis, on the other hand, refers to the production or formation of white blood cells. And next, the megakaryocytopoiesis refers to the production of platelets from megakaryocytes. So, erythropoiesis is influenced by erythropoietin and hypoxia. So, hypoxia meaning low oxygen concentration ang blood. Leukopoiesis are influenced, or the production of leukocytes or WBC are influenced by the presence of colony-stimulating factors and interleukins. So, di ba napansin niyo ganina as we are discussing the leukopoietic progenitors, daghan kayo og mga interleukins na na-mention. And to help you memorize the interleukin or the cytokine that play a major role in that specific leukocyte to mature, natin gihatag na mga mnemonics or palatandaan ng among reviewer sa una. So, si interleukin 5 daw is for eosinophil. Dili siya ingon nga for eosinophil ang gyud si interleukin 5, but si interleukin 5 is the main player in eosinophil maturation. So, para dali daw matiman-an, kay si 5 daw, mura man daw na siya og ibaliktad na E. What? Sorry. 5, kay mura daw og ibaliktad na E. So, kato daw eosinophil for interleukin 5. Next, for basophil, we have interleukin 3. Dali lang po ito. Kay si 3, anak lang, B for basophil. Next, madali lang din si GCSF and MCSF, kaya naalang man sa ilahang pangalan. GCSF is for neutrophil, since si neutrophil man ang pinaka-abundant na granulocyte sa circulation. Now, MCSF is of course for monocytes. So, kato lang, it's so dumb but I hope it helps. And megakaryocytopoiesis is influenced by thrombopoietin and the colony-stimulating factors for megakaryocytes. So, how does hypoxia and erythropoietin play a role in erythropoiesis? So, in cases of anemia, dili lang jud sa anemia, or in cases na hypoxic ang state sa ato ang patient, meaning gamay ang oxygen concentration sa whole system sa ato ang patient, it could be either due to anemia or due to the fact nga nagstay ang ato ang patient sa tag-as na lugar, gamay man, nipis man ang air pressure dito, so gamay lang pod ang concentration sa oxygen. Therefore, medyo hypoxic ang state sa body ana. So, if hypoxic ang state sa ato ang body, ma-detect na no sa mga oxygen sensing mechanisms sa ato ang body. So, ang mga oxygen sensing mechanisms or area sa ato ang body is located in 2 sites. In the kidney, is located in the kidney, specifically in the juxtamedullary cortex, and in the carotid artery. Specifically in the glomus cells of the carotid artery. So, kani sila ang 2 oxygen sensing mechanisms sa ato ang body. So, once they detect that baba na ang mga oxygen level sa blood, they do something. So, si juxtamedullary cortex of the kidney, once they detect na nababa na ang oxygen level sa body, kuhiton niya si peritubular interstitial fibroblast, located also in the kidney. So, si peritubular interstitial fibroblast will produce erythropoietin. Now, si erythropoietin will act upon the progenitors and precursors of erythrocytes to increase production of red blood cells and to prevent the apoptosis of red blood cells, thereby prolonging their life and increasing the number of red blood cells produced. Once increased na ang RBC count sa body, increase na pod ang oxygen carrying capacity. Since, di ba, man gyud na ang main function ni RBC ang mag-carry og oxygen. So, pag daghan na sila, daghan na og cells na mag-carry og oxygen, daghan na og cells na magdakop og oxygen. Therefore, at least man lang, maskin gamay, ma-minimize ang hypoxia. Now, if ma-correct na ang hypoxia, mag-normalize na pod ang rate of erythropoietin production. Kay hypoxia and erythropoietin production is in a negative feedback mechanism. Now, erythropoietin is not only produced in the kidneys. Erythropoietin is also produced or secreted in small amounts sa perisinusoidal cells sa liver. Now, let's proceed to thrombopoietin. Thrombopoietin, just like erythropoietin, will stimulate the production of platelets or will stimulate the maturation of megakaryocytes, therefore stimulating the production of platelets by first inhibiting apoptosis. So, most cytokines na involved sa differentiation or involved sa maturation sa mga blood cells, mo gid na ilahang function no, to prevent apoptosis, to promote the differentiation of cells, and to promote the maturation of cells. Ang unique kay thrombopoietin is that it also promotes endomitosis. So, na-mention na natin ni kaganina, no? Endomitosis mo ni ang unique lang sa mga megakaryoblasts or megakaryocytes. This refers to the division or ang pagdaghan sa number of chromosomes without nuclear division or cytoplasmic division. So, endomitosis, the number of endomitosis is directly proportional to the number of platelets produced. So, if thrice ka nag-undergo og endomitosis, meaning po daw ana, thrice the normal ang imuhang platelet na ma-produce. Thrombopoietin, unlike erythropoietin, nga ma-increase ang production in hypoxic states, si thrombopoietin dili, no? Si thrombopoietin ang iyahang production or synthesis is constant. Thrombopoietin is secreted mainly in the liver or synthesized mainly in the liver. However, napoy small amount sa thrombopoietin nga ginahimo sa kidney. And ang paghimo sa thrombopoietin is constant. Dili jud daw na siya mag-increase or magbaba. So, how is megakaryopoiesis regulated if ang thrombopoietin levels kay constant? Well, first, let's tackle kung unsa ba ang future ni thrombopoietin. So, naay two futures or fates si thrombopoietin. First, it can be taken up by platelets and degraded. So, kauna na sila ni platelets, tapos i-degrade na ni platelet ang thrombopoietin. Or, ang remaining thrombopoietin can go to the bone marrow and stimulate the megakaryocyte to produce more platelets. So, timanin na ha, mauna ang duha ka future ni thrombopoietin. First, it can be degraded by platelets. Or, ang sobrang nga thrombopoietin nga dili ma-degrade ni platelets can be taken up by megakaryocytes. So, if ma-take up siya ni megakaryocyte, dito na siya mag-contribute sa megakaryopoiesis or sa pag-produce pa og more platelets. So, mauna siya, no? Direct ang regulation ni thrombopoietin sa platelet production. Kay, if ever increased ang platelet production or increased ang amount sa platelet sa body, meaning, daghan og platelet nga mag-take in kay thrombopoietin to degrade thrombopoietin. Therefore, gamay na lang ang amount sa thrombopoietin nga mag-adto kay megakaryocyte to promote platelet production. However, if gamay ang platelet count or if gamay ang number sa platelet nga naga-circulate sa body, kana lang, gamay lang, meaning gamay lang pod ang thrombopoietin nga ma-degrade. So, daghan og thrombopoietin nga mag-adto sa ato ang megakaryocyte or megakaryoblast sa bone marrow to promote further production of platelets. So, I hope na visualize ninyo siya, no? If ever nga naagin mo questions or gikalibugan, please do not hesitate to ask questions. I-chat lang ko ninyo. So, again, how does the thrombopoietin regulate megakaryopoiesis? It regulates it directly because of the fact that when platelets are low, when platelets in the circulation are low, gamay lang pod ang thrombopoietin nga ma-degrade, thereby leading to an increased number sa thrombopoietin nga moadto sa bone marrow para i-stimulate ang megakaryoblasts na mag-produce og more platelets. However, if increased na daghan na daan ang platelet count sa circulation, meaning daghan pod og thrombopoietin nga ma-degrade. Then since daghan og thrombopoietin nga ma-degrade, gamay na lang og thrombopoietin ang maadto sa bone marrow to stimulate the megakaryocyte. So gamay na lang ang stimulation sa ato ang megakaryocyte to produce platelets. So inana ang regulation sa megakaryopoiesis ni thrombopoietin. At this point, let's look at the generalized or the very summarized idea of the three types or three subclasses of hematopoiesis. First, the erythropoiesis. So the erythropoiesis occurs in the bone marrow. Diba remember that the specific term for the area wherein the hematopoiesis occurs in the bone marrow is the hematopoietic cords. So if this is the hematopoietic cords, na din hiya ang mga sinusoidal, ang mga endothelial cells na nag-align sa ato ang hematopoietic cords. Erythropoiesis happens near duol sa ato ang endothelial cells. Erythropoiesis happens near the endothelial cells so that dali na lang gud i-release ang mga mature erythrocytes to the circulation. So, para pag mature na ni sila, dali na lang sila mapagawas padulong sa circulation. So, erythropoiesis, na-mention na po nato ang iyahang mga progenitors, beginning from the common myeloid progenitor, si CFU-GEMM. Next, the burst-forming unit erythroid, katong progenitor na wala pa kaayo receptor for erythropoietin. Next, with the action of interleukin-3, GCSF, and other interleukins, burst-forming unit erythroid will become CFUE, katong progenitor na daghan na og receptor for erythropoietin. Now, from the CFUE, the erythrocyte or the erythroid precursors will emerge. So, it is safe to say na si CFUE ang last progenitor stage sa erythrocytes. So, erythropoiesis is influenced or is regulated mainly by erythropoietin. How? Yun nga, erythropoietin will prevent apoptosis, will increase or promote cell differentiation and cell maturation, and it will also induce or it will also promote hemoglobin synthesis. Now, let's proceed to leukopoiesis. Leukopoiesis can be categorized into two, myelopoiesis or lymphopoiesis. Myelopoiesis refers to the maturation and the production of granulocytes. Lymphopoiesis, on the other hand, refers to the maturation and the production of lymphocytes. So, in the bone marrow, if this is the bone marrow hematopoietic cords, immature granulocytes or mga immature granulocytes ato mga progenitor sa ato ang mga granulocytes are located deep within the bone marrow cord. So sa tunga gina sila. But as they mature, maghinay-hinay sila og gawas in order for them to easily go out into the circulation. So in leukopoiesis, daghan jud kayo nga cytokines ang involved, namely GM-CSF, GCSF, MCSF. So na-mention naman ni nato sila no during our discussion about the progenitors and the lineages. If gusto ninyo magbasa more about cytokines involved in hematopoiesis, naa lang siya sa RODAC, any edition, napod siya sa third John, pero sa third John kay mas daghan sa third John. I will not force you or require you or even tell you nga i-familiarize ang tanan nga interleukins because I myself do not memorize them all. I hate memorizing them jud kay daghan jud kay sila. The only thing I am telling you is that kato i-familiarize lang tong mga interleukins na specific or interleukins that play the major role in that specific cell lineage. So, huwag natin pahirapan sarili natin. Kato lang mga important interleukins ang ato ang i-focus or katong mga special na mga interleukins that play the major role in the specific cell lineages. So, kani sila to no? Sila GM-CSF, GCSF, MCSF for the myelopoiesis. Now, si interleukin 3, na-mention ako kaganina nga, siya ang main player sa maturation ni basophil. But even though siya ang main player sa maturation ni basophil, dili siya specific na kay basophil lang. Si interleukin 3, daghan, in fact, almost all blood cell lines, nag-play siya og role in stimulating their blood lineages or their cell lineages like granulocyte, monocyte, megakaryocyte, and erythroid cells. But for basophil maturation and differentiation, si interleukin 3 gyud ang nag-play og main role. Nag-play pod si interleukin 3 og side role for granulocyte, monocyte, megakaryocyte, and erythroid cell maturation. For lymphopoiesis, the B-cell maturation and B-cell proliferation is regulated mainly by B-cell growth factor, and for the T-cell is interleukin 2. Megakaryopoiesis, on the other hand, occurs dito lang yun na siya solely, unlike sa leukopoiesis nga mag-start siya sa center or sa deep area sa hematopoietic cords. Megakaryopoiesis happens only in the dili lang jud na area near the sinuses or near the endothelial cells of the hematopoietic cords. Same reason sa erythropoiesis, so that dali lang niya ma-release ang platelets into the circulation. Kaya diba remember, platelets are from megakaryocytes. Si megakaryocytes, sila ang pinakadako na cell sa bone marrow. They do not leave the bone marrow kaya di sila kagawas tungod sa ilahang kadako. But ginapagawas nila ang ilahang platelet na na-produce through ginapalusot nila ang ilahang cytoplasm sa mga sinusoidal pores or endothelial pores na nag-align sa ato ang hematopoietic cords for the platelets to go out to the circulation. So, kung ano si megakaryocyte, gina-extend niya ang iyahang cytoplasm para ma-release si platelet. So, megakaryopoiesis, yun nga, happens in the area near the vascular lining of the hematopoietic cords. Now, there are many cytokines involved in megakaryopoiesis. No, actually, all of them, erythropoiesis, leukopoiesis, megakaryopoiesis, many cytokines are involved. So, dili na lang nato na sila isa-isa huntanan, but let's just focus on the fact that the main cytokine, the main hormone needed for megakaryopoiesis is the TPO or the thrombopoietin, which is mainly produced by the liver and is secreted or synthesized in small amounts in the kidney. Now, let's proceed to the principles of normal cell maturation. Dali lang ito, no? Always bear in mind that in cell maturation, mas daghan og work jud ang mga immature cells because the immature cells will prepare everything that is needed by the mature cells in order for it to do its work. So, pag mag-ingon ta nga mas active, mas busy, mas daghan og activities na ginahimo ang immature cells, therefore ma-imagine dayon nato nga ang mga cellular characteristics sa immature cells kay katong cellular characteristics sa very active na cell. So, asa na yung mga gipang mention, ma'am? For example, the overall cell size. Pag immature ang cell, mas dako siya. So, as the cell matures, the cell size will also decrease. So, nganong mas dako man ang cell size sa mga mature na cell? That is because daghan og organelles present, since daghan og organelles nga naga-work for the preparation of the cell in maturation. Another thing is that the nuclear cytoplasmic ratio is also increased in immature cells, but the nuclear cytoplasmic ratio is decreased in mature cells. Therefore, the nuclear cytoplasmic ratio decreases as the cell matures. So, what is this nuclear to cytoplasmic ratio? NC ratio refers to the amount of space occupied by the nucleus in relationship or in contrary or in comparison to the space occupied by the cytoplasm. So nuclear cytoplasmic ratio decreases as the cell matures because the nucleus size also decreases as the cell matures. So for example, ito no, this is an immature cell. Dako ang iyang cytoplasm, dako po ang iyang nucleus. Ngano ganito? That is because more active ang immature cell's nucleus kay naga-prepare man siya sa mga organelle, sa mga proteins nga needed by the mature cell in the future to do its work. So again, presong gi-mention ganina, mas busy si immature cell kay mas busy siya in preparing the cell to do its function kapag mature na sila. So for example, no, kay mga erythrocytes. So mga immature erythrocytes are busy in preparing itself para mahimo niya ang iyahang function sa future, which is to deliver oxygen. So ang ginakabisihan sa mga immature erythrocytes kay mag-produce og hemoglobin. Kay para inigdako niya, mature na siya nga erythrocyte, na siya enough amount of hemoglobin nga needed niya in order for it to do its work. So again, let's go back to NC ratio. So if this is the cell's cytoplasm, dako po siya no, and this is the cell's nucleus, mapansin nato nga almost all of the cell cytoplasm is occupied by the nucleus. However, as the cell matures, maghinay-hinay og kagamay ang iyahang nucleus, ug maghinay-hinay pod og kagamay ang cytoplasm. However, dili proportional ang rate sa ilahang paggamay. Mas dako ang rate sa paggamay sa nucleus kaysa sa rate sa paggamay sa cytoplasm. Therefore, leading to the decrease in NC ratio. Kay mas nagamay man ang nucleus kaysa sa paggamay sa cytoplasm. Therefore, as the cell matures, mas gamay ang space nga ma-occupy ni nucleus sa whole cell. Or mas gamay ang space na ma-occupy ni nucleus kaysa sa cytoplasm. What are the nuclear changes that happens in our cells as it matures? First, naay changes mahitabo sa chromatin pattern, sa nuclear shape, nuclear size, and the presence of nucleoli. Chromatin pattern. So in immature cells, ang chromatin pattern is smooth, homogeneous, fine, lacy parehas ninyo, og nako, joke lang, dili ta lacy. Delicate and thready. So in general terms, immature cells chromatin pattern are loose or dili pa sila inana ka-condensed. Why? Because the immature cells, ang mga chromatin nila are very active pa. Therefore, dili isa gyud dapat sila condensed. Di ba you have learned in your cytogenetics na ang chromatin, if condensed sila kaayo, meaning inactive na sila. So in immature cells, wala pa kaayoy inactive chromatin because most chromatin function pagyud in preparation for the cell maturation. But in mature cells, some chromatin will become condensed, will become clumped, or in fact pycnotic. Grabe na gyud kaayo ka dense or compact. Like this. Ginani ang pycnotic na chromatin, pycnotic na nucleus no. Kanang dark na gyud kaayo ang chromatin. Kay very condensed na jud sila. So, nganong nagapa-condensed ang chromatin pattern as the cell matures? That is because some of the chromatin will become non-functional na. So, i-fold na sila or ginapa-condense na sila og taman kay dili naman sila needed. Nagamit naman sila in the earlier stages of the cell's life. Next is the nuclear shape. So, immature cells have round or oval nucleus. Nuclear shape as a parameter for determining the cell's age or maturity is useful lang no for WBCs. That is because si WBCs, sila lang man to ang naay changeable na nucleus. Ang RBCs, kay constant man yun na nga circle lang yun ang ilang nucleus. So again, As the cell matures, ang iyahang nuclear shape will become more segmented or more lobed. That is actually owed to the same reason nga mas active ang immature cells or mas naa sila'y nuclear activity. Bantog circular gyud ang ilahang nucleus. But as the cell matures, naay mga parts sa nucleus nga mahimo ng filaments or magnipis na naay mga parts sa nucleus, di ba? If immature pang cells, oval na siya. But as the cell matures, naay mga folding na nga mahitabo. Like, from the oval, kay mahimo na siyang inana. Naay mga lobes na ma-form. Again, that is owed to the fact that as the cell matures, some parts of the nucleus will become non-functional. Therefore, mas ginapa-compress sila, mas gina-fold. Therefore, mas mahimong segmented or lobed ang nucleus. Next, the nuclear size. Same reason again, due to the fact na si immature cells kay mas active, mas nagapangailangan og nuclear activity, the nuclear size of immature cells are large. But as the cell matures, manggamay ang iyahang nuclear size, kay mag-diminish naman pod ang nuclear activity as the cell matures. Next, in RBCs, di ba, ang makita na tong RBCs sa circulation are anucleated, meaning they do not have nucleus. That is because RBC's nucleus are extruded or ginapagawas na siya sa cytoplasm sa RBC at one stage of its maturation. So, kana pod, isa pod sa determinant sa cell maturity, especially sa mga erythrocytes, is the extrusion of its nucleus. As the erythrocytes mature, hinay-hinay na pod na nga ginapagawas ang nucleus or hinay-hinay na ginapa-prepare ang nucleus for extrusion or pagpagawas sa nucleus. Next, the presence of nucleoli. So, immature cells do have nucleoli or nucleolus, whereas in mature cells, the nucleoli are not visible anymore. The changes we have mentioned earlier are the nuclear changes. There are also cytoplasmic changes that will happen in our cells as it matures. First is the cytoplasmic basophilia. The cytoplasmic staining characteristics of our cell will also change. Immature cells tend to have bluer cytoplasm because it contains more ribosomes. And diba, they have more ribosome kay as what I have mentioned earlier, mas active ang ang immature cells kay gina-prepare niya ang iyang self sa iyahang work na iyahang himuon as it will mature. So, if ang mga immature cells have more ribosomes, and please recall that ribosomes are made up of RNAs, ribonucleic acid, ang mga RNAs will attract the basic dye. So, most basic dyes are color blue. Therefore, since daghan